Hi everyone, welcome to lesson 3.2, Prokaryotes. Make sure to fill out your guided notes and keep up with your vocabulary as we go through this lesson. So first we're going to have a quick review. Okay, remember we have prokaryotes and eukaryotes. We learned these two terms on our last lesson. Those were vocabulary terms last lesson. But prokaryotes are cells that do not enclose DNA in a nucleus, and eukaryotes are cells that do enclose DNA in a nucleus. And if you remember last time, animal and plant cells were considered eukaryotic, and next lesson, that's whenever we kind of go into the details of those two. But today we're going to focus on prokaryotic cells. So if you recall, prokaryotes are unicellular organisms that lack a nucleus. They have DNA, but the DNA is not found in a membrane-bound nucleus, but in the cytoplasm instead. And for many years, prokaryotes were synonymous with bacteria, but now we classify them a little differently. So biologists have divided prokaryotes into two distinct taxonomic groups, bacteria and archaea. Right now you don't have to memorize this table, you're going to see this when we go through our evolution unit. But here's a nice breakdown of how we categorize things. So there are three domains of life, bacteria, archaea, and eukarya. Bacteria and archaea are prokaryotic, while eukarya is eukaryotic. Then you can see within those domains we have kingdoms. So the domain bacteria corresponds to the kingdom eubacteria, while the domain archaea corresponds to the kingdom Archaebacteria. So here are two types that we're going to talk about today, bacteria and archaea, and you notice that they both have definitions here. So peptidoglycan is a polymer of sugars and amino acids that surrounds the cell membrane. Bacteria are a domain of prokaryotes that contain peptidoglycan in their cell walls. And remember, bacteria live almost everywhere, freshwater, saltwater, land, within human bodies, and within other eukaryotes. And some are good, some are bad. And I put those in quotation marks because, that's how we like to classify things. So even though bacteria don't have the best reputation, again, bacteria, some are considered good and are very important within ecosystems. Archaea are a domain of prokaryotes without peptidoglycan in their cell walls. And although archaea and bacteria look very similar to each other, the DNA sequence of some key archaea genes are actually closer to eukaryotes than bacteria. And most archaea live in extremely harsh environments. So here's a video back in Blend. Go ahead, pause here, watch this video, answer those two questions, and come back whenever you're done. Okay, let's go ahead and move on to bacterial structure. So prokaryotes vary in their size and shape, in the way that they move, and in the way they obtain and release energy. The typical bacterial structure can be seen here. So whenever we see bacteria, this is often kind of that structure that you'll see labeled in your textbook. So bacteria is the plural term, and bacterium is the singular term. So remember, DNA is free floating. So all this blue, this blue kind of spaghetti looking stuff, that is the DNA. The ribosomes are all of these little dots here. So all those kind of brown dots are ribosomes, and they synthesize proteins. The cell wall shapes, supports, and protects the cell. And remember, for bacteria, the cell wall does contain peptidoglycan. The cell membrane regulates materials entering and leaving the cell, and it also protects and supports the cell. The pili or pili, depending on where you're from, how you say it, the pili are short hair-like structures. So those are all these little structures here on the cell surface of prokaryotic cells. They can have a role in movement, but really they're most associated with adherence to surfaces. And then last, the flagella or flagellum, depending on singular or plural, those are used primarily for cell movement. So we're going to go into bacterial structure a little bit more in detail. So prokaryotes can range from 1 to 5 micrometers and come in a variety of shapes. We have bacilli, which are rod-shaped. So kind of this is that bacilli shape rod, or kind of look like a pill shaped. We have cocci, which are spherical. And then we have spirilla, which are spiral and corkscrew shapes. Some prokaryotes do not move. Some are propelled by flagella. Some glide through a slime-like substance they secrete. So again, bacteria are very diverse, but we do try to classify them based on their size, shape, and if they can move. So next is their arrangement. This is some extension information. You don't have to necessarily know this, but it is interesting. So bacteria can also be classified on how they are arranged. So diplo is an arrangement of 2 cells. Strepto is a chain of cells. And Staphylo is a cluster of cells. So that's often how we end up naming bacteria, is both based on their structure as well as their shape. So a common method of distinguishing between types of bacteria is through the use of a Gram stain. So remember, all bacteria contain peptidoglycan in their cell walls. Gram staining differentiates bacteria by the chemical and physical properties of their cell walls. So gram-positive cells have a thick layer of peptidoglycan in their cell wall, and it retains the primary stain crystal violet. So crystal violet is the stain that we use for this bacteria. Gram-negative cells have a thinner peptidoglycan layer that allows crystal violet to wash out, and therefore they will appear pink. So this is really kind of the information you need to know here. Gram-positive bacteria are purple, and gram-negative bacteria are pink. And prokaryotes also need a supply of chemical energy, which they can store in the form of fuel molecules such as sugars. Energy is released during cellular respiration or fermentation, much like humans. So prokaryotes are very diverse with regard to obtaining and releasing energy. So here's kind of that table. So prokaryotes can be heterotrophs, photoheterotrophs, photoautotrophs, or chemoautotrophs. Additionally, prokaryotes can live with or without oxygen. We call that living with oxygen or needing oxygen aerobes. If they do not need oxygen, we call them anaerobes. And some can survive in either condition, and we call those facultative anaerobes. So when a prokaryote has grown to nearly double its size, it replicates its DNA and divides in half. And that's going to produce two identical cells, known in a process that we call binary fission. So binary fission is a type of asexual reproduction in which an organism replicates its DNA and divides in half, producing two identical daughter cells. And again, it's just called asexual reproduction because that cell is dividing and you don't require any mixture of genetic information. When growth conditions are favorable, prokaryotes can grow and divide very quickly, some as often as once every 20 minutes. When growth conditions are not favorable, many prokaryotic cells form what we call an endospore. And an endospore is a thick internal wall that encloses DNA in a portion of the cytoplasm. So endospores can remain dormant for months or even years, and this is one of the reasons why many prokaryotes can survive in harsh conditions. So then the question that we always get is, well, since bacteria and prokaryotes can reproduce asexually, how can they evolve? So there's two ways in which that can happen. The first one is mutations, and mutations can be inherited by daughter cells produced in binary fission. We're going to talk about this in evolution once we get there, but again, if it's a favorable mutation that helps that bacteria survive, then that favorable condition can be passed along to the next generation, and that's kind of one of those things that drives adaptation. The next thing that we have is called conjugation, and that's the process in which prokaryotes exchange genetic information. So right here, this is that this is that binary fission happening, okay, that cell dividing. This is conjugation, okay, where it's showing the connection, and then this one right here is the endospore. So in your textbook, if you want to look up this photo, it kind of explains that in better detail. But again, that endospore helps those prokaryotes to remain dormant for months or years and can survive in harsh environments. So let's look here at this conjugation. So conjugation, a hollow bridge forms between two bacterial cells, and genetic material, usually in the form of what we call a plasmid, moves from one cell to another. So in bacteria, a plasmid is a small circular DNA strand, and the transfer of genetic information increases genetic diversity in populations of prokaryotes. So whenever we get to genetics for Unit 4, then we're going to talk about bacterial replication, okay, and we're going to learn that term plasmid more. But for now, again, a plasmid is just a small circular DNA strand. So the importance of prokaryotes. Prokaryotes are essential in maintaining every aspect of the ecological balance in the living world. Some species have specific uses in human industries as well. So we've talked about this a lot in Unit 1. I think that we've seen this before, but remember, prokaryotes can be decomposers, they can be producers, they can fix nitrogen, and many of them have human uses, and we use them for a wide variety of food and other commercial products. So prokaryotes are extremely important for ecological balance. So last we're going to talk about those quote-unquote bad bacteria. So remember, prokaryotes are extremely important, and even though bacteria often have that negative connotation associated with like something being dirty, bacteria are everywhere and oftentimes very beneficial and helpful to the environment. So here are a few of the diseases caused by bacteria. Disease-causing agents are called pathogens, and bacterial and viral pathogens are the most common. Currently, there is no known pathogenic archaea. Some bacteria can disrupt health and cause diseases by destroying living cells or releasing chemicals that upset homeostasis. So, basically, two ways in which they do that. They can damage host tissue or they can release toxins. If they damage the host tissue, it can cause tissue damage when they provoke a response from the immune system. Some can release toxins that can act as poison, which interferes with the normal activity of the host. And down here is just kind of a table of a few different bacterial pathogens. So here's our exit ticket. Go ahead, pause here, and try to answer this on your own. Okay, so the two domains for prokaryotes were bacteria and archaea. Here is our true or false. Okay, prokaryotes contain DNA, but they do not contain DNA in a nucleus. Prokaryotes have ribosomes, and remember, bacteria did have peptidoglycan in their cell walls. That's why the statement is false. And then all prokaryotes were unicellular. Binary fission was a type of asexual reproduction in which an organism replicates its DNA and divides in half, producing two identical daughter cells. Conjugation was the process in which some prokaryotes exchange genetic information. And the last one, do all bacteria cause disease? No. Remember, bacteria are also essential in maintaining ecological balance. They can be decomposers, nitrogen fixers, and producers. Bacteria that cause disease are called pathogens or pathogenic bacteria. So that is the end of lesson 3.2. Go ahead, review your notes, and thank you very much for watching.